We're going to look in this video at what happens in the proximal convoluted tubule. So here I've drawn a diagram of the nephron, and here's the Bowman's capsule where the actual filtration takes place. This is the proximal convoluted tubule. This is the distal convoluted tubule. This is the collecting duct, okay, and here, of course, is the loop of Henle. Right, so let's have a look at what happens in the proximal tubule, which is really important. An awful lot of the processing of the filtrate occurs here. Okay, so what happens, we won't talk about how it does it just yet, but you get about 60% of the water in the filtrate is taken out and back, it goes back into the blood, essentially, which is essential, of course, otherwise you'd be, the kidney filters, 100 centimeters cubed every minute. You'd be losing 100 centimeter cubes of water every minute if you didn't take some of that back. 60% of the sodium ions, they're also reabsorbed, and all of the glucose and all of the amino acids, 100% of them are taken from the filtrate back into the blood. Obviously, you don't want to excrete them. So let's have a look at the mechanism by which this occurs. Okay, so I just move this down here. Right, what we've got here is an epithelial cell. We've got epithelial cells which line the kidney tubule, so the nephron. So what we've got here are on this side is the filtrate, that's there, and here we've got the interstitium. Okay, so there's more cells there, and of course somewhere over here you are going to have blood vessels. You're going to have a capillary. Let's see if I haven't drawn to the same scale. But so the idea is we've got to get all of the sodium, the water, the glucose and so forth out of the filtrate, across the epithelial cells and into the capillary. That's what's got to happen here. Okay, so let's see how this is done. Now the whole thing that is driving this here is the ATPase, sodium potassium ATPase pump, which you should be familiar with from the nervous system. Okay, it's present in all cells. It plays a really key role in reabsorbing stuff from the filtrate. So if you remember, for every 1 molecule of ATP which is hydrolyzed, the energy from that is used to pump against the concentration gradient. You get 3 sodium ions will be pumped out of the cell, and 2 potassium ions will be pumped into the cell. So as a consequence of that, what's going to happen is you're going to get a low sodium concentration inside the cell. And so what happens is you've got a higher sodium concentration here. So sodium ions should be able to just diffuse through, okay, into the cell. Okay, and once it's in the cell, then it gets pumped out the other side, and eventually this is going to make its way into the capillary. It's not quite as simple as that, because what also happens here is it's not just simply the sodium passes down a channel. There's a specific carrier molecule here, a co-transporter, and what it does is it will transfer 1 sodium ion into the cell, and at the same time that's down the concentration gradient. Going against the concentration gradient, it's going to take a glucose across. So if you like, the exothermic process of sodium ions diffusing into the cell is used to drive the endothermic process of getting glucose into the cell, because the glucose is going against the concentration gradient. And in that way, all of the glucose comes out of the filtrate and makes its way into the cell, the epithelial cell. And then on this side of the membrane, you have channels, and the glucose can simply diffuse out there. But if you realize what's happening there, then really the whole thing that is driving this is the ATPase pump, with the energy from the ATP is using to pump out the sodiums, and the low sodium concentration here is used to transfer the glucose across. Also, exactly the same thing happens, very similar thing happens with amino acids. You get a co-transporter, so you get sodium ions coming in, and every time you get one sodium ion, you get an amino acid molecule coming in as well there, and that eventually will pass, through. It will come out here and into the blood. Also, you probably hasn't escaped you notice that the potassium ion concentration is going to increase as that ATPase pump is working all the time. And what happens there, you have a channel here for potassium to come out. Okay. Interestingly, there aren't any potassium channels on this side of the filtrate side, on the lumen side. So you don't lose potassium into the filtrate. It all ends up goes in this direction when it diffuses out. Okay, so in that way, let's talk about the water as well. If you are transporting sodium and glucose and amino acids, these are all osmotically active substances across, they are going to cause water to come across here. So you're going to get H2O coming there from the lumen into the epithelial cell, and then across that side of the epithelial cell into the interstitium. Water is going to do that as well. But the whole thing is driven by the activity of the ATPase. Okay, so in the next video, we're going to talk about what happens to the loop of Henle.